Today, we're going to be diving into genetic engineering by talking about how we can work and manipulate DNA. But first, as always, I want you to go and mark yourself present for attendance. You should have already reviewed this PowerPoint document, all 9 slides. And before you start, I want you to go up to the top, click View, and show speaker's notes. I want you to start doing this in all of my PowerPoints now, Google Slides documents. Because on many of my slides, I will have important notes that will be helpful in addition to the slides. So let's begin working with DNA. In science, we're now able to cut, separate, and replicate DNA base by base. Remember, our bases are A, T, G, and C. A binds with T, and G binds with C, and vice versa. Now scientists are able to read DNA's base sequences for any organism. And let's talk about cutting DNA. So DNA, we know, is just way too large to be analyzed in one piece. So we need to cut it up into segments to analyze it. And how do we do this? We do this by restriction enzymes. Restriction enzymes are enzymes that simply cut DNA at specific base sequences. There are thousands of restriction enzymes, and each one of them recognizes a certain sequence of CT AGs, all right, different bases, different combinations, and they cut them there, right? We see the enzyme, and it cuts the DNA. So let's stop and check and examine this question. The diagram below represents a laboratory process. We see a picture, DNA is being cut with what we'll call scissors. So the substance represented by the scissors shown cutting DNA is 1. An enzyme, 2. A starch molecule, 3. A carbohydrate, or 4. A fat molecule. So the answer is choice 1. The scissors represent an enzyme. More specifically, they represent a restriction enzyme, cutting DNA at a specific base sequence. Now we've cut all this DNA. How do we separate it? How do we sort the DNA? Well, this is done by gel electrophoresis, a lab method used to separate DNA fragments. Now let's expand upon this. Gel electrophoresis separates DNA fragments by size, and it uses an electric current, right? You can see a current running into this machine, into the gel. Larger molecules travel a shorter distance, and small molecules travel a longer distance. So, we load DNA into the gel. We have a marker which indicates a known size of base pairs, but we have different samples, and we're able to see the sizes are base pairs that the DNA travels through the gel. Remember, larger molecules travel the shorter distance, and smaller travel longer. So this is the basic idea of DNA fingerprinting, right? These gels will show that no two organisms' DNA is the same, well, and perhaps unless they're twins or a clone. And yes, so the marker serves as a reference point. Let's stop and examine this. So base your answers to the following question on the diagram below and your knowledge of biology. The diagram shows the results of a technique used to analyze DNA. The technique used to analyze DNA directly results in A. Synthesizing large fragments of DNA, B. Separating DNA fragments on the basis of size, C. producing genetically engineered DNA molecules, or D, removing the larger DNA fragments from the samples? The correct answer is B. Gel electrophoresis is used to separate DNA fragments on the basis of size. So how can we use DNA? We're going to talk about and go into detail into bioinformatics, studies that assemble and maintain databases of human DNA. So we're going to talk about genomics, study of whole genomes. And right, remember genomes are all the genetic material contained in an organism. So in genomics, we'll talk about individual genes and their functions. And genomics can be used to study many different species. And what's interesting is the Human Genome Project, which was started in 1990 and completed their task in 2003. Their job was to map entire sets of DNA for organisms. They wanted to map an entire human genome, all the genetic material in a human. So that took 13 years and $1 billion. But now if you want to map the entire genome of a human being, it takes roughly 1 to 2 days and costs $3,000 to $5,000. So you really can see how science has advanced. In further talking about genomics, we're going to dive into how genomics leads the development of technology for medicine and farming, genetic testing, right? We can now test if we have a higher susceptibility to certain diseases. We can even use this to treat diseases like diabetes and cancer. And yeah, we can sequence somebody's whole entire genome to pinpoint what might be wrong with them and to find the right treatment for them. And we're going to be discussing this further in tomorrow's lecture. So in ending, just remember, everyone's genome is different. Our genome is like our fingerprints.